So in this clip we will discuss about the inheritance, basics of inheritance, some basic terms like chromosomes, DNA, gene, genome. What is the difference between these terms? Nucleotides, the structure of nucleotides, and RNAs, and about messenger RNA and its role in the synthesis of protein. First of all, the most familiar terms in the topic of inheritance are chromosomes, DNA, and gene. These all stand for similar meanings like the genetic coded information. Like our cells, they always carry some information when they divide. So this information is transferred to the new cells. So the DNAs, they carry this information in form of codes. So all the cells, they follow the instructions given in the code, and accordingly these cells divide and make new cells. So in simple, inheritance is a topic in which we study that how the DNA and genes, they give some coded information to cells to divide, to specialize, and to make some specific type of proteins. So this is a sequence. Like we have a nucleus inside the cell. So inside the nucleus there are chromosomes, and chromosomes are made up of DNA, deoxyribonucleic acid, and a part of DNA is called gene. So gene is basically an inheritary unit which carry a specific character in an organism. So gene is a part of DNA. So what is our DNA? DNA stands for deoxyribonucleic acid, and these are the polymers of nucleotides. Polymers of nucleotides. So nucleotide is a monomer, a basic unit which repeat itself and make a DNA. So polymer mean nucleotides repeat itself to make a DNA, or to make another version which is called RNA, that is also a polymer of a nucleotide. So a nucleotide consists of three components. One is called phosphate group. You can see this is phosphate group, and the other one is pentose sugar. In the middle is a pentose sugar, and the third one is called base or nitrogenous base, and this one is a base. So phosphate group, pentose sugar, and nitrogenous base. So each nucleotide can have one of the four bases. Four bases are A, adenine, guanine, C for cytosine, T for thymine. So but one nucleotide will have only one of these. So you can see this is one nucleotide, and this is another nucleotide, and this is another nucleotide. So and onward. So all these nucleotides together, they make a chain, they make a polymer, and that polymer is called DNA. So DNA is is a double helical structure. You can see there are two structures which make a DNA. Two strands twisted around each other, so they make a DNA. So, and here you can make a notice that phosphate, pentose, sugar, and then again phosphate, then pentose, sugar, then again phosphate. So this part is called the backbone of the DNA. So that is called phosphate and pentose sugar backbone. So they make a backbone of the DNA, and we will have on the other side we will have nitrogenous bases. So there is another term, complementary base pairing. So these are four. So one nucleotide will have either adenine, guanine, or cytosine, thymine. So on the other side, so adenine, if this one is a adenine, so that will always make a pair with thymine on the other side. So thymine on the other side, making two hydrogen bonds in between. And if it is guanine over here, so that will always make on the other side with cytosine. Okay, so this one, that A will make a bond with T, or T will make a bond with A. So these are specific. And guanine, G will make a bond with C, or C will make a bond with G. So these are specific, means never it can happen that T can make a bond with G, or A can make a bond with C. So this is called complementary base pairing, means these bases are complementary to each other. So on the other side, so they will make a bond. The other nucleotide, if this nucleotide is having adenine base, so the other nucleotide on the opposite side should have a base of thymine. Okay, so this is one strand, on the other side there is gonna be another strand, and these two strands together, like you can see the structure, two strands are twisted around each other by these hydrogen bonds, so they will make a DNA molecule. And so what is a gene? Gene is basically a small part of DNA. So each gene control one character, one inherited character in an organism. And there is another similar type of term which is called genome. Genome is called the entire set of genes in any organism. Like if we say human genome, so it means the entire set of genes inside the humans is called human genome. So recently, a few years back, the scientists, they have studied the human genome, and as a result now it is more easier to study them, the inherited disorders, and to find an evolutionary evidence by finding and by studying the human genome. Okay, so evolutionary evidence means finding out the tiny differences between the different races of human beings on the planet. So RNA is another version of DNA. DNA is always have two strands, and RNA is also a polymer of a nucleotide. Nucleotide is having a similar structure consisting of three parts: phosphate group, pentose sugar, nitrogenous base. So the same nucleotides, when they make a single strand like this, so they are called RNAs. We have 2-3 different types of RNAs, transfer RNAs, messenger RNAs, ribosomal RNAs. But here we will study about mRNAs, which stands for messenger RNAs. So messenger RNAs basically carry a message from DNA to ribosomes. So DNA are located inside the nucleus, but they are too big to come out of the nucleus. So it means they carry the genetic information, the coded information, but they are not able to come out of this nucleus. But the ribosomes, where the protein synthesis process happens, is outside of the nucleus, inside the cytoplasm. So it means ribosomes will make proteins getting the genetic information from DNA, but because DNA cannot come to ribosomes, so messenger RNA will come to DNA, will carry the coded information and will carry this information from DNA to ribosomes. So messenger RNA is a coded version of a part of DNA, so that will carry information that will come to DNA, because that is smaller, that can cross the nucleus membrane. So that will come in, copy some information from the DNA and will go back to ribosomes and will leave that information over there, and according to that information the ribosomes will arrange the amino acids in a specific sequence, and according to that sequence the protein molecules are made, and the specific sequence will make specific type of protein molecules. I guess this is an all for this topic, and I hope you will understand this topic. Thank you. Bye.